Welcome to Chapter 7, Microbial Biochemistry, Section 5, Using Biochemistry to Identify Microorganisms. Learning objectives. Describe examples of biosynthesis products within a cell that can be detected to identify bacteria. Accurate identification of bacterial isolates is essential in a clinical microbiology laboratory because the results often inform decisions about treatments that directly affect patient outcomes. For example, cases of food poisoning require accurate identification of the causative agent so that physicians can prescribe appropriate treatment. Likewise, it is important to accurately identify the causative pathogen during an outbreak of disease so that the appropriate strategies can be employed to contain the epidemic. There are many ways to detect, characterize, and identify microorganisms. Some methods rely on phenotypic biochemical characteristics, while others use genotypic identification. The biochemical characteristics of a bacterium provide many traits that are useful for classification and identification. Analyzing the nutritional and metabolic capabilities of the bacterial isolate is a common approach for determining the genus and species of the bacterium. Some of the most important metabolic pathways that bacteria use to survive will be discussed in microbial metabolism, which is Chapter 8. In this section, we will discuss a few methods that use biochemical characteristics to identify microorganisms. Some microorganisms store certain compounds as granules within their cytoplasm, and the contents of these granules can be used for identification purposes. For example, polybeta hydroxybutyrate, PHB, is a carbon and energy storage compound found in some non-fluorescent bacteria of the genus Pseudomonas. Different species within this genus can be classified by the presence or absence of PHB and fluorescent pigments. The human pathogen Pseudomonas aeruginosa and the plant pathogen Pseudomonas syringae are two examples of fluorescent Pseudomonas species that do not accumulate PHB granules. Other systems rely on biochemical characteristics to identify microorganisms by their biochemical reactions, such as carbon utilization and other metabolic tests. In small laboratory settings or in teaching laboratories, those assays are carried out using a limited number of test tubes. However, more modern systems, such as the one developed by Biolog Incorporated, are based on panels of biochemical reactions performed simultaneously and analyzed by software. Biolog's system identifies cells based on their ability to metabolize certain biochemicals and on their physiological properties, including pH and chemical sensitivity. It uses all major classes of biochemicals in its analysis. Identification can be performed manually or with semi or fully automated instruments. Another automated system that identifies microorganisms by determining the specimen's mass spectrum and then comparing it to a database that contains known mass spectra for thousands of microorganisms. This method is based on matrix-assisted laser desorption ionization time-of-flight mass spectrometry, MALDI-TOF, and uses disposable MALDI plates on which the microorganism is mixed with a specialized matrix reagent. The sample reagent mixture is irradiated with a high-intensity pulsed ultraviolet laser, resulting in the ejection of gaseous ions generated from the various chemical constituents of the microorganism. These gaseous ions are collected and accelerated through the mass spectrometer, with ions traveling at a velocity determined by their mass-to-charge ratio, m to z, thus reaching the detector at different times. A plot of detector signal versus mz yields a mass spectrum for the organism that is uniquely related to its biochemical composition. Comparison of the mass spectrometry to a library of reference spectra obtained from identical analyses of known microorganisms permits identification of the unknown microbe. Microbes can also be identified by measuring their unique lipid profiles. As we have learned, fatty acids of lipids can vary in chain length, presence or absence of double bonds, and number of double bonds, hydroxyl groups, branches, and rings. To identify a microbe by its lipid composition, the fatty acids present in their membranes are analyzed. A common biochemical analysis used for this purpose is a technique used in clinical, public health, and food laboratories. It relies on detecting unique differences in fatty acids and is called fatty acid methyl ester analysis, or FAME. So of course I can't say FAME without thinking of the song. You're mostly probably too young for that reference. In a FAME analysis, fatty acids are extracted from the membranes of microorganisms, chemically altered to form volatile methyl esters, and analyzed by gas chromatography, GC. The resulting GC chromatogram is compared with reference chromatograms in a database containing data for thousands of bacterial isolates to identify the unknown microorganisms. A related method for microorganism identification is called phospholipid-derived fatty acid analysis, PLFA. Membranes are mostly composed of phospholipids, which can be saponified, hydrolyzed with alkali, to release the fatty acids. The resulting fatty acid mixture is then subjected to FAME analysis, and the measured lipid profiles can be compared with those of known microorganisms to identify the unknown microorganism. Bacterial identification can also be based on the proteins produced under specific growth conditions within the human body. These types of identification procedures are called proteomic analysis. To perform proteomic analysis, proteins from the pathogen are first separated by high-pressure liquid chromatography, HPLC, and the collected fractions are then digested to yield smaller peptide fragments. These peptides are identified by mass spectrometry and compared with those of known microorganisms to identify the unknown microorganism in the original specimen. Microorganisms can also be identified by the carbohydrates attached to proteins, the glycoproteins, in the plasma membrane or cell wall. Antibodies and other carbohydrate-binding proteins can attach to specific carbohydrates on cell surfaces, causing the cells to clump together. Serological tests, such as the Lancefield groups tests, which are used for the identification of Streptococcus species, are performed to detect the unique carbohydrates located on the surface of the cell. Oof, that's a lot of text, sorry. I'm going to read it to you. Clinical focus, resolution. Penny stopped using her new sunscreen and applied the corticosteroid cream to her rash as directed. However, after several days, her rash had not improved and actually seemed to be getting worse. She made a follow-up appointment with her doctor, who observed a bumpy red rash and pus-filled blisters around hair follicles. There's a figure for that. Wait, what? Ah, there it is. Oh, doesn't look that bad. Okay. The rash was especially concentrated in the areas that would have been covered by a swimsuit. After some questioning, Penny told the physician that she had recently attended a pool party and spent some time in a hot tub. In light of this new information, the doctor suspected a case of hot tub rash, an infection frequently caused by the bacterium Pseudomonas aeruginosa, an opportunistic pathogen that can thrive in hot tubs and swimming pools, especially when the water is not sufficiently chlorinated. Pseudomonas aeruginosa is the same bacterium that is associated with infections in the lungs of patients with cystic fibrosis. The doctor collected a specimen from Penny's rash to be sent to the clinical microbiology lab. Confirmatory tests were carried out to distinguish Pseudomonas aeruginosa from enteric pathogens that can also be present in pool and hot tub water. The test included the production of the blue-green pigment piocyanin on centromide agar and growth at 42 degrees Celsius. Centromide is a selective agent that inhibits the growth of other species of microbial flora and also enhances the production of Pseudomonas aeruginosa pigment piocyanin and fluorescin, which are a characteristic blue-green and yellow-green, respectively. Tests confirmed the presence of Pseudomonas aeruginosa in Penny's skin sample, but the doctor decided not to prescribe an antibiotic. Even though Pseudomonas aeruginosa is a bacterium, Pseudomonas species are generally resistant to many antibiotics. Luckily, skin infections like Penny's are usually self-limiting. The rash typically lasts about 2 weeks and resolves on its own, with or without medical treatment. The doctor advised Penny to wait it out and keep using the corticosteroid cream. The cream will not kill Pseudomonas aeruginosa on Penny's skin, but it should calm her rash and minimize the itching by suppressing her body's inflammatory response to the bacteria. And that brings us to the end of Chapter 7. Next time, it'll be Chapter 8, Microbial Metabolism. Until then.